Brilliant. Hi, I have to say, it's genuinely a real pleasure to be here and giving this lecture. Thank you very much for the kind introduction, Gillian. I'm going to talk to you today about, essentially to talk all about cancer, as I'm sure you can see from the title. I'm going to start off with an introduction to the disease, but then move on through particularly research that we've been doing, and my group and I have been doing for something like 10 or 15 years, really trying to understand more about the molecular mechanisms, the real details, the nitty-gritty of what goes wrong and changes a normal cell into a potentially aggressive cancer cell, and how we can use that information to develop new drugs and improve treatments. And then near the end, I'm just going to have a couple of quick comments on novel technology projects that we would never have started if we hadn't moved to Heriot-Watt here and found really, fantastic new collaborators, particularly bioengineers working in microfluidics, which I'll comment on very briefly, and in 3D bioprinting. So to start on, there's always that thing of make sure you make your information in your talk relevant to your audience. Unfortunately, that's not really an issue here, since the numbers are in the UK now, approximately half of us are going to have a diagnosis of cancer at some stage of our lives. This number's been increasing over recent past, and that's really because we have an increasing lifespan, and cancer being a disease of old age, this is, it arises through the accumulation of damage and mutation in our normal cells as we get older. So fundamentally, why do we get cancer? It really just comes down to being kind of an inherent problem of multicellularity. If you're an E. coli cell, whenever you get food, you grow and you divide as much as you can until you run out of food, then you try and stay alive until there's more food around. Our cells, we have trillions of cells, and each one of our cells contains and retains the capability to grow and proliferate and make more cells. That being the case, multicellularity has meant that we've had to evolve really reliable, robust systems to make sure that our cells only grow and divide when they are instructed to, and when it's in the best interests of our whole, for things like wound repair and regeneration. So this issue of having trillions of cells also means, though, that if we have trillions of cells that all have the capability to divide, there are trillions of each represents an opportunity or a possibility that those control systems will go wrong, and therefore this, the development of hyperproliferative damaged cells is really just an unavoidable problem of having of our large multicellular nature. So I'm going to start off with this. It's actually quite a new diagram, but it's an old sort of idea. This is a kind of classical view of tumor development that was around when I was starting to think about cancer research. And this is really that we have normal cells. Here's in an epithelium, a surface layer of cells, and they accumulate some mutations. An initiating first mutation that increases proliferation, causes storage to an increasing number of cells in a clone carrying that mutation, and then you get subsequent changes until, maybe 5 or 10 changes might occur that will lead to a heterogeneous, multi-clonal population of cancer cells that have the potential to be a lethal disease. The point here being, these mutations in all of our genes are happening all the time, but there are only a few that affect cancer, and the only ones we really pick up are those that ended up leading to an aggressive clone of cells that was hyperproliferative and produced a really big lump that you went to the doctor with. And how does this affect our healthy status in patients? These are survival rates for the common types of cancers in the UK these days. Some are not a terrible diagnosis. Things like testicular cancer, melanoma, and breast are relatively treatable. But there are things like brain tumors, lung cancers, and pancreatic tumors that are essentially untreatable, and the survival of which is extremely unusual beyond a year or two. But the difference here really is a couple of things to say. One is that these tumors up here, the apparently good ones, will all kill the patient if left untreated. And it's really our ability to identify these diseases early, or the fortunate circumstances of biology and humanity that you were likely to notice these tumors earlier, and our ability to treat them, rather than these that are diagnosed tend to be seen later and are impossible to treat with current therapies. So this point here, cancer treatment for many years has really relied on radiotherapy and cell-killing chemotherapies that are effective treatments for some types of cancer, but have little selectivity for cancer cells over normal cells, and they will kill our healthy dividing cells and therefore lead to the side effects we all know of things like hair falling out and sickness from the killing of the intestinal lining and those kind of things. But those side effects limit the amount of drug we can give, and therefore the ability to use these drugs to successfully treat many types of cancer. Therefore, for the last at least 30 years or so, the core strategy in cancer research has been to identify these specific molecular changes that are driving a particular cancer, and therefore that are converting that normal cell into an aggressive, potentially lethal cancer cell. To identify those specific changes and try and interfere with those, so you find the changes that are driving the cancerous state, and particularly those on which the tumor remains reliant, or are, for example, stopping it being, causing it to evade immune detection and destruction by our immune systems, and develop drugs to reverse those changes and to treat the cancer. And therefore, the hope being that those would have far fewer side effects and could be given in higher doses and be more effective therapies. Sometimes this really, really works. So this is a really good example. I've heard it described as the poster child for targeted therapy. But this is imatinib, which was discovered 15 or 20 years ago and has really changed the prognosis for chronic myeloid leukemia patients, from this being something that was almost always fatal to being a disease that is manageable and sometimes curable. When we wanted to think about these, so our question is that these changes that we want to get at, what are they? We define the mutations that drive cancer into two general groups. Oncogenes and tumor suppressors. An oncogene is something the function of which, when it is increased, makes cancer more likely. So it's essentially a kind of cancer driver gene, and the proteins encoded by oncogenes. And the converse are tumor suppressor genes, and the proteins those genes encode. The point there is that the tumor suppressors are active in our normal cells all the time, and yet when their functions happen to make cancer less likely, and therefore when they are damaged by whatever changes, whether by deletion of the whole gene or a mutation, after all we are subject to continual mutation. If tumor suppressor genes are damaged, then cancer becomes more likely, and so most and we've been cataloging these changes, particularly over the last 10 years or so. The glioblastoma is an aggressive brain tumor, and it's first of these was published about 10 years ago. Since then, there have been many what I would call cancer genome studies, in which large consortia of researchers, clinical and molecular, have been cataloging the changes that you identify if you take 1,000 breast cancers, or 300 or 400 brain tumors in this case, and you look at the mutations that those many different patients have, and the changes in gene expression and behavior of those cells that occur in those cancers. And what they found was, firstly, that this is such an important point, every cancer is different. So you can, if you have 1,000 breast cancers, you can find molecular differences between every one of those. And that's probably the main hurdle in cancer treatment, is that there is absolutely not one size that fits all. The other point, though, is that there are very much the usual suspects. So there are sets of proteins that work together in functional units in our cells, and I'll show you in one or two of those in detail in a moment, that act together, and we can cluster them in groups here and say that, well, this protein acts on this protein, and if this one's damaged, then there's no point damaging the, you find these things are changed in many, many tumors. And therefore, if we're developing drugs, the best places to go for are these sets of proteins that are changed in many, and in some cases, most, and almost all human cancers in some cases. So, so one of these happens to be something I've spent a lot of my academic career studying, and there's a thing called the PI3 kinase signaling pathway. Now, I don't want to go into it in too much detail and with loads of jargon, but nearly all of our cells have their growth and proliferation controlled, as I said at the beginning, by external signals. They only grow and proliferate when they're told to, and these signals come in the form of things called often growth factors, and all of our cells have different proteins on their surfaces that receive these incoming signals that tell them to grow. And it's this cell receiving and interpretation systems that we call signaling pathways that are damaged in nearly every human cancer. And an example here is this PI3 kinase pathway. So nearly all of the receptors that tell our cells to grow share the characteristic that when they are stimulated and switched on, they activate and turn on an enzyme that's in all our cells called PI3 kinase. And this thing is, as I say, it's an enzyme. It's a catalyst for a chemical reaction. And it adds a phosphate group onto one lipid, and it makes a little bit of another lipid that's a growth signal. And nearly everybody just calls that PIP3. And when the levels of this lipid go up, a cell is more likely to grow, to proliferate, and to perform kind of what we anabolic metabolism, bring things in and build more cell. And it does this, it causes these downstream changes through a set of set of proteins. There are at least 20 of these, and there might be as many as 100. We're still not quite sure how many things bind to this lipid and can be controlled by it. One of these is called AKT, and I'll be talking about that a tiny bit for at some stage in the next 10 or 15 minutes. And that's clearly a really important signal itself. When it gets switched on, it makes cells grow and proliferate. But there are many others, and we don't understand the importance of the others very well in our cancers. The thing I've been working on, though, is this enzyme here called PTEN, which all it does is get rid of PIP3, and therefore it's a classic tumor suppressor. If we all have, every cell has 2 copies of every gene, so we have 2 copies of PTEN. If your PTEN gene or genes get damaged, you lose the function here, and the levels of the growth signaling lipid increase, and therefore cancer is more likely to occur from that cell than when PTEN was normal. And therefore, it's damaged in about a third. We find that this is damaged in about a third of all human cancers. And the reason we're interested in the real nitty-gritty details is we need to understand this. We can't literally put PTEN back into all the cells in a tumor. That's practically and kind of technologically impossible. But we can develop drugs to block things like AKT. But knowing what to develop your drug against is the key question at the beginning of every kind of drug development, drug discovery project. So the things my lab's been spending a long time on are the questions I'll very briefly comment on. We did a lot of work to try and understand how the enzyme is controlled, to see if that would open up new pathways to treatment. I'm going to say a little bit about what we can learn from PTEN mutations in inherited patients. But this key question here is, what downstream of PI3 kinase and PTEN, what's driving the tumors, and are our drug development projects have they got the right targets? So I just want to start here. PTEN, as I said, is an enzyme. It's got 403 kind of amino acid building blocks that fold up like this to make an enzyme that catalyzes that change in that metabolism of the lipid. It is itself has the activity as an enzyme controlled by chemical modifications of various sorts. And over the years, we've studied a few of these. Like many proteins, it's controlled by the addition of phosphate groups here. This is essentially inhibitory to PTEN. Again, I'm sorry, I'm going to show you some real, genuine data in a minute, but this is just still sort of summary stuff. PTEN also gets controlled by reversible oxidation, so the actual active site of the enzyme can have its active site nucleophile oxidized, which switches that off, but it can be recovered by reduction. And finally, you get inhibited by what's called ubiquitination. And ubiquitin is a little, as it may surprise, very common protein unit that is around all our cells, gets physically attached to PTEN and most other proteins. And in the case of PTEN, this directly inhibits the enzyme, but it also targets PTEN for destruction, which is actually the best appreciated, understood function for PTEN. So to get up, but so I really wanted to put out, largely because of all the people who've worked an awful lot to do that research, and it seems a shame to. We can talk about it another time. But so what I want to talk about is show you a first bit of data from inherited mutations in PTEN. So I've been talking about sporadic tumors that occur essentially at random. But a few unfortunate individuals also inherit one damaged copy of PTEN. So we all have 2 copies of all our genes. Patients who inherit one damaged copy of PTEN have a variety of symptoms, but they're very, very diverse, and they're very hard to predict. So some patients, as you might expect from what we've been saying, that patients inheriting a broken copy of the one PTEN gene have a dramatically increased risk of malignant cancer. But they also have many of them have a range of developmental problems of sort of benign lumps and bumps. But there are other patients who have very different symptoms, who have, say, autism spectrum disorder and macrocephaly, which is essentially a big head, but don't have the other problems that are common in these mutation carriers. And what wasn't clear for a long time, it took 10 or 15 years to start trying to understand, is there a link between these specific mutations that arise in the gene and are affecting, specific, for example, amino acids along here, just changing one of them? And this is a diagram of where those mutations have been found in inherited patients. Is there a link between the specific mutation and the symptoms that are arising in the patients carrying those mutations? Now, it seems bonkers biology if there wasn't, but actually understanding that has been really, really hard. So I just want to talk about here, for Laura Spinelli in the lab, I think they did some really nice work. We got on the front cover of the Journal of Medical Genetics a few years ago with this point here, and this is a kind of lab workhorse experiment, so I'll talk through it in a little bit of detail. We take some PTEN, some cells from a tumor patient, actually they've been around, grown in laboratories for the last 50 years or so that don't have any PTEN. So they have, I talked about AKT as being activated by PTEN here. So these cells here, if you don't do anything with them, they've got no PTEN, and they've got very high activity of AKT because there's no PTEN. And that's revealed by this black blob in our blotting experiments. But here, if we then use the PTEN gene, we put it in a virus, and we add increasing amounts of the PTEN gene. We can increase the amount of PTEN here. This is shown by the increase in the staining here in these Western blots. And as you get more and more PTEN, you get less and less activity of AKT. So this is active functional enzyme that makes this black signal from AKT go away. This is wild, that WT stands for wild type, so healthy PTEN, putting healthy PTEN here down with AKT. If we have a broken version, though, this is the classic inactive mutant version of our enzyme, C124S, we increase the amount of PTEN, and absolutely nothing happens, because the enzyme can't be active, it can't metabolize its lipids, so it has no effect on the cells. But what Laura found when she took 7 different mutants that have been identified in the sequencing from autism spectrum disorder patients was that these are patients whose phenotype is certainly caused by their PTEN mutation. And yet, really surprisingly, when she made these viruses and put them into the cells, she found in every case, she found that as you increase the amount of PTEN, you got a suppression of AKT. And therefore, this shows us that these mutant proteins expressed in this specific group of patients actually are capable of being perfectly active. They're very unstable proteins in some cases and are kind of falling apart and not doing their job as well as they should. But this was a real surprise, and it therefore suggests that this partial activity may be related to, it sounds dangerous and to describe autism spectrum disorder as a milder phenotype, but compared to some of the more, what we would say, the more severe patients with numerous tumors, I would argue that's the case. But it's certainly a distinct phenotype. And the important thing here is this may allow us for the first time to start predicting for those patients, when kids come in the clinic and they're diagnosed with a PTEN mutation, at the moment, or until recently, we've had no way of saying, we think this is the likely outcome for you, for you and your family over the next 20 or 30 years. And this ability potentially to predict phenotype is potentially really, really good for that patient group. And this has been carried on by Virginia Alvarez Garcia, who's here. And Virginia's work has really shown that we weren't barking up the wrong tree. Her data firmly agree with this, and we're trying to take this forward and make it more useful. So the next few minutes, I'd want to talk briefly about experiments to understand, as I mentioned, how exactly is PTEN suppressing tumors? Why is this important? This is important because the pharmaceutical industry, even 10 years ago, had provided us with an awful lot of drugs. And they're very good drugs at doing what they were designed to do, of getting into our cells and suppressing the activity of the targets they were designed to inhibit. However, very few of them have so far proved to be really good cancer therapies. And that's probably because we didn't understand the biology of the cancer well enough before we started those drug programs. A few of them, Cal-101, however, for example, is a kind of fantastic a success and is a real kind of wonder drug for B-cell derived lymphomas. It's great, but only for a very small number of patients. But how can we understand that question of which are the important things that are something like PTEN that can do a lot? Which are the important things downstream that are driving the tumor phenotype? But we have a real resource for that of the mutations in the cancer patients. So here's a plot of the position along the gene again from 0 to 403 in the amino acids of the PTEN protein, and the number of mutations you find. So they're spread out all over the place. Most of these mutations that arise in the patients, however, are very uninformative in that regard. They just kill it stone dead, and they don't help by allowing you to connect specific downstream effects with the development of a tumor. So what we want are, and we looked for quite a long time, were mutations that had only lost one or two of the many things PTEN can do, in order that we could then say this one's connecting with the tumor phenotype. This is probably the function that's important in tumor suppression, and that's where we should be designing our new drugs. But through a lot of hard work, a few members of the lab, again Laura particularly, but also Fred, Lindsay, and Priyanka, identified a pretty large group of some mutations, so about 11 different mutations in different regions of the protein that shared this characteristic that they could all, and I'm not going to explain all the experiments in too much detail, but they all were active in a test tube, so you could make the protein and dephosphorylate the lipid. If you put them into cells, they made the levels of the lipid go down. And if you test the activity status of AKT again, which most people had been considering as the main mechanism of activity, of tumor promotion when you have a PTEN mutation, all of these things were able to keep AKT inactive and unphosphorylated in contrast to these genuinely dead mutants here. So this implies that there must be something else that is connecting with tumor formation. So that, again, just to show those are in different bits of the protein. And we also wanted to test, sorry to whiz through some of this data, we really went to town on a few of these to see whether we weren't missing some subtle failure to control AKT and kind of what we'd call canonical AKT signal, canonical PI3 kinase signaling, and we can't find anything. So these curves and these data all overlap, between our mutants and the normal healthy protein. So this bit of what they can do looks normal. But we did find a couple of cell-based assays. And the point, so these data here, everybody focus, we and everybody else focus on AKT because this is so easy to do. Doing a lot of experiments, doing things like these three-dimensional cell culture-based assays, however, is much more time-consuming and difficult, but gave us a much better correlation between the data we saw and the tumor mutation, the point that these mutations were arising in cancer patients. So these are epithelial cells grown in three-dimensional protein matrices that are quite like they'd find in the body. And a few types of these cells here, these MDCKs and NMUMGs, if you seed them, will proliferate and form a small colony of cells that has a hollow center surrounding a hollow lumen, much like they would around a tubule or a duct in the body. But we found that this absolutely required PTEN. If you got rid of PTEN, they just failed to organize themselves, and this architecture was just deranged and wrong. And in contrast to the connection with AKT, with all these mutants that I've just shown you here, all failed in this assay here, making a much, and the same with experiments in which we looked at brain tumor cells and their ability to invade in 3 dimensions through these matrices. So so these more complex cell-based assays gave us a much better correlation with the apparent information from the patients, but that's still not really good enough because you can't go to a cancer conference and call this cancer. So this is, again, so we then collaborated with Doreen Cantrell's lab, and this is the only time we've made a genetically modified mouse for our work, is to, and in this case, so we introduced 2 of these mutations into lines of genetically modified mice. And if we have, and I'm not going to go into the details too much, but if you can make cells from these mice that express only the mutant proteins here, this thing Y138L. Cells that have no PTEN have, again, it's the same sort of test of looking at the activity of AKT using these antibodies. If you have no PTEN, you have a really high activity of AKT, but even just one copy of this mutant protein is enough to keep the normal pathway in AKT here low and in its normal state. So this is hyperactive and but deranged, this is normal. And we also found, though, that if we crossed the mice, we couldn't, despite the point that this mutant protein looks like it's keeping the standard pathway down, we still couldn't get any live births of mice that had 2 copies of the mutant protein. So you need 1 healthy copy to have a mouse. And the key experiments here were that if we looked at tumor formation in these mice, again with Doreen and Stuart, who's a pathologist, if we allow a group of mice to age, the healthy mice get a few tumors when they get quite old. This is age along here and percentage of mice sort of tumor-free. But this has been done for a long time, as if you have mice with 1 healthy copy and essentially 1 completely broken, missing copy or inactive copy of PTEN. These mice come down with tumors. In this background, it's almost all B-cell lymphomas. They get these tumors around a year of age, 1 year of age. But our mice with this partially functional mutant copy, this Y138L, their tumor development is much delayed, and after all, that is correlating with these things have a hyperactive AKT and these don't. But the point is here, there's something going on in those mice, despite their apparently normal signaling, if we analyze it using the standard approaches, that is causing the tumor formation in pretty much every single one of that group of mice. And I think that's the really important conclusion, therefore, is that in these mice, there is something other than AKT that is being activated downstream of PTEN that's required for the viability of the mice. And it's also correlating with the formation of all of these tumors. And therefore, this, I'd argue, crudely speaking, this is where we should be, we need to look for other pathways downstream of PTEN for where our new drug development projects should be looking. So, yeah, and I probably haven't got to, again, so therefore we are developing new tools. This is a collaboration with Rory's lab. We need to find out more about these other pathways downstream. So we, given the time, I should probably move on. But again, so we're developing sort of fluorescent probes that we can put into cells to study in more detail these localized signaling events, because what's almost certainly the case is the reason we study AKT, because it's really easy to do and you can smash open your cells and look in biochemistry. This is much harder to do here. But these fluorescent probes using microscopy can show you where specific sort of populations of a protein are regulated in a cell, even if most of the protein in the cell is not being changed. So anyway, I might be able to answer questions. But so now I want to really change tack quite a lot for the last 10 minutes or so. and just talk about a couple of project areas that we would never have got into if we hadn't moved our lab here about 5 years ago, with a couple of bioengineers here at Heriot-Watt. The first is some work collaborating with Maiwen Kazodi-Karawaz, who's a microfluidic bioengineer who has technologies to use microfluidics to purify first plasma from blood and then DNA from that plasma. And why would we be interested in this? The point there is for a cancer biologist, we all have a little bit of DNA floating around our circulation. So in every mil of all of our blood, there are a few thousand copies of our genome floating around in a maybe free state, probably associated with some proteins. But remember, so that's a few thousand copies of the genome. We have, don't forget that mil of blood contains millions of white blood cells and billions of red blood cells. So there's not a lot of DNA there. But in a cancer patient, after all, in some, or in some, many other disease, people with disease burden, some of that DNA is coming from the tumor. And that provides the really, really exciting possibility that we might be able to learn about the tumor by taking a blood sample rather than by having to book them into hospital for an invasive operation that sticks a needle into some piece of their body to physically sample the tumor tissue. And therefore, the point there is that might be hard to use to detect and to screen for tumors. That's something people talk about. But it's definitely going to be good for characterizing a tumor that you know is there. And it's almost certainly going to be very useful for monitoring disease burden over time. For example, if a tumor's been resected in a surgery, if you take a blood sample every few months, can you start picking it up again a few months later? Because in most cases, that method appears to be more sensitive than most scanning methods. And finally, if you give a drug and have a treatment, is somebody responding to therapy? Does their tumor DNA start to disappear as their tumor regresses? And if you can do all of that with blood samples rather than much more invasive, expensive things, then that has to be good for everybody. So again, this is mostly based on Maiwen's technology, and I'm not going to show you any real data other than this image from Virginia's recent paper. But this is very much a project underway, but we're trying to use collaborate and combine microfluidics with kind of standard molecular biology to see if we can enrich the genes that we'd be interested in to analyze them. And we also, the Virginia side project here was to develop really simple assays that can be hopefully sensitive enough to still pick up these rare markers in blood. And Virginia and Eva, who is sitting next to her, are combining a few different experimental approaches to try and get that to work. So I'm now going to, for the last few minutes, finally, again, on another technology issue. Remember, we come back to this diagram here of a classical model of a tumor. So this is how people tended to think about cancer 10 or 20 years ago. And the point is, it has been drawn just as a big blob of cancer cells. Unfortunately, the reality, if we look at lots of clinical data, is that what we term the tumor microenvironment in a real patient is way more complex than that. So the cancer cells in a tumor will be very heterogeneous and will contain many different clones with many different lineages of accumulated mutations. But there will also be numerous more healthy cell types that we tend to call stromal cells that have infiltrated into the tumor. So some of those are immune cells that should be, you hope, mounting an anti-tumor immune response to kill the tumor. But tumors also evolve the ability to recruit cells in that provide a supportive role and keep the tumor proliferating and alive. And there are also other things like connective tissue cells and cells that make new blood vessels that are all within this tumor milieu. And if we want to do experiments to study cancer research, we have to be much close to this rather than just having a blob of cancer cells or some cancer cells just growing on a plastic dish in our culture suite. So Some sad news here is this point that's been, but although very old, this is the probability of success of new drugs through clinical trials. And you'll see the point here. So some disease areas, like infectious diseases, for example, are better performing, and the key numbers are the ones on the main line here. So more than 15% of new drugs that go into a patient for the first time end up 5 years later, maybe, or 6 years later, being approved for use in widespread medicine. Cancer, unfortunately, is consistently the worst performing area here, and it's probably due to that heterogeneity that every patient is different. But the number here that's slightly blacked out is 7%. So from 2014 data, 7% of new drugs that go into phase 1 clinical trials, only 7% of those get approved. And the point there is that, so 93% of those, the evidence that we had at the beginning, the preclinical experiments and all that research was not giving us a decent informative set of data on which we could make that decision to invest in clinical trials, which are extremely expensive in terms of financial and human cost. So I think this is a really nice illustration of how much we need better experimental models to both to study the disease and also to test drugs for safety and efficacy. So if we want better disease models, what are our choices? If we want to replicate really complex biology, this is a slice of the small intestine, we can work with animals, but there are so many reasons why we don't want to do that. Not only the obvious ethical issues, but they aren't particularly great at predicting success. There are lots of cancer research studies making the point that we've been able to cure cancer in mice for decades, but the complexity, and there are many reasons for that. But so more recently, so if we want to try and engineer cell culture-based experiments, we now have really, really good ways of preparing cells with just the right genetics. We have gene editing technologies and so on, and we have induced pluripotent stem cell technologies that allow us to take cells with the right genetics, human cells, and program their state of differentiation to turn them into, for example, a liver cell if we want a liver, or a cardiac cell if we want to work on the heart. But if what we then have to do with them is just grow them either on a plastic dish or as a blob of cells suspended in some jelly, that we know that they don't behave the way they would in the body. And what can we do to improve this? One of the things that the sorts of ways of improving this appeared as an opportunity when I arrived here. It took me a little while to realize that 3 doors down in the corridor was a bioengineer called Will Shue, who was building bioprinters, 3D bioprinters. But after a while, we started collaborating, and unfortunately, Will has now moved to Strathclyde, but he got a professorship. I couldn't argue, but so the strategy here is to take cells, and I'm sure you're all aware of the ideas of 3D printing, is to layer by layer build up solid object, 3-dimensional solid objects through the sequential deposition of different layers to build up structures. And that if you have a complex 3D printer, you can have multiple lines to build up 3-dimensionally complex objects using different components in different locations. So the point here is that we can combine different types of cell that we would find in a tumor with appropriate matrices, and there's a lot in that statement. So we want whatever's around those cells, we want that to be both representative of what the cell will experience in the body, but we also have, for the 3D printing process, we have real demands in terms of its mechanical properties. And what we end up doing, certainly in our project, is using some stuff from seaweed called alginate, which has the fantastically convenient property that it's a polysaccharide, but the strands of the polymer become cross-linked with each other when they're exposed to divalent cations like calcium or barium, and therefore you can have it fluid in your printer and then exposed to calcium and have a dramatic, very fast increase in mechanical rigidity and stiffness that allows you to build up a self-supporting structure. And the whole process takes minutes, up to an hour, depending on the complexity of your construct. So I'm going to talk just a few things here. So we started doing this with Will in his lab-built printers. More recently, we've been very fortunate to collaborate with Marcus Ardren at Renishaw, who's here. And this has been, I think, I hope he would agree, a really mutually beneficial collaboration on the grounds that Marcus's printer is considerably more reliable when the person who built it is not in the room than Will's printers were, I think understandably. And this has 6 delivery lines that allow us to build up pretty complex structures. And just to go through a couple of the things we've done here, we don't have a lot of data. This project's clearly midway through. But the alginate on its own isn't really recognized by the cells. So if you just print cells in alginate, these are a couple of different types of brain tumor cells and macrophages. They just kind of sit there. They're alive. They'll proliferate, but they're not really. This isn't how the body looks in vivo normally. But so we modify that matrix to ensure that the cells will engage with the matrix itself. And we also incorporate a few other proteins and a few other components that we find in the body in the normal extracellular matrices. And the cells behave much more as they would and form things that are much more tissue-like. And the resolution, by which I mean the smallest thing we can print, is clearly a massive feature here if we're thinking about what we can do with this technology. We can't yet kind of print a liver or a kidney with that, positioning a cell here, the next cell here, the next cell here, to make a tubule around in the exceptionally complex 3-dimensional structure of our real complex organs. We can print things with a resolution of around a few hundred microns and keep all our cells alive and so on, which isn't very good if your aim is to try and print a liver. But if you want to print something with the spatial sort of size, scale range differences that are around in tumors, the spatial non-uniformities there, this is a really useful capability. So what we've ended up doing. Are printing, initially the first thing is to print really crude structures like this, usually something like a core of pure tumor cells in the middle, surrounded by a layer which has both tumor cells and more healthy stromal cells that we have identified, and we've got from clinical collaborators that have been purified from patients' tumors. And we can print a set of these pretty reproducibly, and then we can, for example, use them to test drugs against. So this is just some data from Miguel. And again, I'm not going to go into the details here, but just to say that this is as good a drug as you can get for a brain tumor, which, to be honest, isn't very good, but it is approved, and it provides a small benefit for the patients. But the point here is we get a pretty similar sensitivity to the drug killing the cells in the 2-dimensional adherent culture as we do in our printed 3-dimensional cultures. But if we go for something like cisplatin, which the cells are pretty sensitive to in 2-dimensional culture, and based on that and success in some other tumor types, this was tested in clinical trials for brain tumors on several occasions. What we get is that we see that in 3 dimensions, our brain tumor cells are highly resistant to this drug when they are exposed in a 3-dimensional setting. Again, that's been shown by other people before, but it agrees very nicely with the data that have come back from the clinic. And therefore, it would have been nice to have this data 20 years ago and maybe have avoided some of the clinical trials. So just to finish off there with this, so again, we need to understand what our printing does a bit more, but this point here is a big challenge is to incorporate something like blood vessels into those printed constructs. And I'm sure you can all think through the difficulty of that when we're adding cells layer by layer in a 3D printer. But in the medium to long term, I think that I really, really like these projects, because I think too many of this, if we're thinking about more complex, better experimental models, too many of the models I've seen over the years for 3-dimensional culture are very bespoke and they're very complicated. So you need a really good postdoc to come in and spend 2 years developing a new model of skin or melanoma. And if that, a bit like the 3D printer, if that person's there doing the experiment, they can get it to work really well. But transferring that capability to a new lab is in practice very difficult. And it's certainly hard to have one person doing that in the morning and then something completely different with some different types of cell and different biology in the afternoon. Whereas I think the hardware for the 3D printers, I think might end up being quite expensive, tens of thousands of pounds, possibly a few £100,000 for a really all singing, all dancing setup. But that's an awful lot cheaper than a mouse house, you know, developing an animal research facility. And is frankly something we'd be, you know, that with human cells, if we can get this to work better, it's something we'd really want to do. But I think that has the potential flexibility once the things are set up with the 3D design files and interpretation files. You can have somebody come in the morning, you could have a postdoc or a facility manager print your brain tumor construct, a dish of 96 brain tumor constructs in the morning, and come back in the afternoon and have the machine run and print a set of melanomas, and the next day you can be doing some cartilage. And I think there's real legs in this as a flexible technology. So I'm going to finish there, and obviously the main thing is really just to thank the many people over the years who it's been a real pleasure to work with, and maybe who've been my own supervisors and mentors over the years, both here in Heriot-Watt here, the people who are currently around in Bowles, and back in Dundee when I was there for quite a few years, and many collaborators it's been a pleasure to work with. Are importantly our funders, and the very last thing is to thank other people who've supported me, and particularly my mum and dad are here. I think Mum took the photo, and my dad's here. Anyway, thank you very much.